Hey, this is Dr. Rob. Welcome to Biblical Genetics. Today I'm walking along a trail near my house, getting some exercise, looking at beautiful springtime flowers and trees and seeing all these people biking and running and stuff. I love getting out here. This is one of my favorite little exercise tracks near my house, but many, many miles on this road. But the topic today is about me getting too deep into a topic and getting way over my head of something that I should never have actually started. You ever do one of those things, or you have an idea and you get started on this idea and pretty soon you realize it's a lot bigger than you thought? Or you had a project and you realize it's gonna take a lot longer than you thought? We had a joke at Georgia Tech. It was like, you estimate how long it's gonna take you to do something, double it, and then go to the next time increment. So, it'll just take me a couple of minutes. Well, it's gonna take you a couple hours. That's just a running gag. It never really worked, but it's kind of what we always did. So I asked myself a question. The question was, hey, what is the longest piece of DNA shared between humans and chimpanzees? Simple question, not easy to answer. And I have spent months on this. In fact, I'm not even finished. I don't know the actual answer, but I'm going to be able to give you a roundabout answer, an approximation after looking at millions of letters of DNA. But how do you think you might go about answering that question? If I said, here's two different genomes, find the longest match, what would you do? Well, you could do it the ridiculous way, like start at the beginning of the human genome, the first letter's an A. Scan through the chimpanzee genome, find the A. Okay, it's in letter 3. So that tells you there's a match with one letter. What about 2-letter matches? Well, start with the first 2 letters of human genome, AA. Scan down the chimpanzee genome. I think you'll find it at like letter 12 or something like that. So you know there's a 2-letter match. Go back to the beginning, take a 3-letter match. First 3 letters of the human genome, you gotta go like, I don't know, position 137 or something like that in the chimpanzee genome. Go back to the beginning. Let's look at 4 letters. Pretty soon, you're going to be scanning through the entire chimpanzee genome and not finding a 7 or 8 or 9 letter match. Going back to the beginning and starting at position 2 and scanning through, oh, there's my 9-letter match. Okay, go back to the beginning. Now you're position 2, and now you start a 10-letter match. You go to the end, go to position 3, go to the end, go to position 4. Oh, there's one. You know, that is ridiculous and slow. No one wants to do that. You'll never, ever finish doing it that way. So I wrote a little computer program to do that. And sure enough, it gets bogged down exponentially quickly. Instantly finds a match of 1, 2, 3, 4, and then pretty soon it's scanning through the entire chimpanzee genome looking for a long match. So that didn't work. So what's next? What else might we do? Well, we could do something crazy like make a dictionary of all the combinations of letters in the human genome and their position. Or opposite, let's do it in the chimpanzee genome. Let's take the whole chimpanzee genome. We'll look for every like 15-letter combination, every single one, the whole genome. Make a giant table of all the 15-letter combinations and all the positions in the genome. Yeah, I tried that just to see, and I actually was able to show that if you broke it down into 12-letter sections, 13-letter section, 14-letter, 15, how many rows and columns it's going to create. And again, that is an exponential problem. The more letters you have in your dictionary, the less places you find it, but the more rows you have. The fewer letters you have in your dictionary, the fewer rows, but the more places you'll find them, so the more columns you have. And that data table is massive. And I'm not a data processing scientist of any way. I wouldn't know how to do it efficiently. So I gave up on that too. You know, I did try for a little while. They said, wait a second, there's a computer program. It's called BLAST. We've talked about it a lot on this channel. The human-chimpanzee argument has gone back and forth over the last several months, mosquito, looking at this program called BLAST. Well, that's what this program does. It'll make a dictionary or a database of all these letter combinations. I believe, well, the way I usually do it, I use a default, and that's 11 letters. It'll make a dictionary of all the 11 letter combinations in a genome. Then when you want to search for something in that genome, like, you know, take a piece of human, 1,000 letters long, it'll break that up into 11-letter chunks, find those positions in the chimpanzee genome, and that's a seed. It'll start there and try to extend. Okay, so you can do that. Problem is, BLAST isn't really optimized for doing what I want to do, and it's a lot of searches. So maybe I can make a giant alignment of the human and chimpanzee genomes. Okay, so there are things, lots of different things. One of them is called last-Z. And so I get on the last-Z documentation, I start looking it up, start learning how to do it, exploring how to start this. And well, I quickly realized that I don't have the computing power. In fact, it says in the documentation, for large genomes, you're going to need a computer cluster. Yeah, okay, I'm not going to do cloud computing for something like that. Oh, but wait, the UCSD, University of California, Santa Cruz, they have a massive genomic center, and they had a last-Z alignment of the human and chimpanzee genomes. Not the most up-to-date genomes, but an older one. So I said, what if I just grab the alignment data? Maybe I can look through there and find out where the longest pieces are. So I did. I downloaded the whole thing. I started looking at it, analyzing it. What's this file format? How's this stuff stored? And pretty quickly I realized that the data is useless. Yeah, it'll tell you matches. It'll tell you all the matches. This one's 1 letter long, this one's 100,000 letters long, 50,000 letters, 10,000, 3. All these different lengths of matches all over the place. And it will tell you how many gaps are in that match. So if you have a 50,000 letter piece of the chimpanzee genome, yeah, it might line up with the human genome, but you have to chop up and add spaces. It doesn't tell you where the spaces are. It'll tell you the number of spaces, but not their positions. So you can't know the length of the matches. So, okay, that's worthless. That was a lot of work for nothing. And then I said, okay, let's look at another alignment program, one like Mummer4. Well, that was great, I thought, and he would have given me the data I wanted, but first of all, it only runs on Linux. And yeah, I've done Linux before, but it's been a long time. And I do have an installation of Linux on my system, and yeah, I could do it. It's just frustrating because I'm not skilled in Linux. It's a lot of work to work on that system, even though I know, all data scientists understand it much better than me. But also, it's dependent on Perl, an old computer language. Computer language not even updated anymore. Now, I am proficient in Perl. I programmed in Perl for 15, almost 20 years, and I loved it, except I don't have it installed on my computer right now. I don't want to. I want to move away from Perl and go to Python. That's what I've been doing for a year now. I just didn't, I don't know, am I being lazy? Or maybe I'm actually being a little smart and saying, no, if I go that route, it'll be even a deeper rabbit hole than I'm already in, because I'm already weeks into this project and I haven't started generating any data. So what am I going to do? How am I going to figure this out? Well, I went back to BLAST again. This computer program is really easy to run once you get it going. You can automate it and it'll just find matches, and it can do all sorts of different ways to find matches. What I realized was if I set it to, oh, little buggy. If I set it to only give me perfect matches, no 98%, no 95%, just 100% matches. And I'm looking at perfect matches between human and chimpanzee. And if I set it so that there are no gaps, it'll skip over any place where you have to add a gap for an insertion or deletion, well, then I'm getting a perfect match. So I started doing it, and I started throwing 1,000-letter chunks of the human genome at the chimpanzee genome. And I did a lot of them. I did like 5 million letters probably over a couple of weeks. And I started getting a lot of data back. But then I realized, oh, wait a minute. No. It'll tell me that there is a match in this 1,000-letter segment, but it doesn't tell me where. Like, what if there's a 10,000-letter match between humans and chimpanzee, and I just happened to pick the very beginning, and the first half is not aligned, and the second half is where the alignment is. Well, that means the alignment keeps on going. So I would have to start there and start counting beyond that 1,000 letters that I gave it. But what if I lined it up on the end of an alignment? Well, that means that I have to count backwards going upstream. So I did figure that out. I wrote a really long computer program. I worked really hard on this. And it was slow because, I'm not working in C or anything like that. I'm still Python's a pretty slow language still. But I also realized I could do it a lot faster. So once again, deeper down at the rabbit hole, I said, wait a second. The data that I'm using, brand new genomes, full length, no gaps, no strange letters. I mean, usually in genomic data, there's mistakes or uncertainties. And there's usually a lot more than 4 letters. You can have A, C, G, and T, sure, but then the letter N represents we don't know. And in fact, the original genomes had like 10,000 Ns, 100,000 Ns in a row. That just represented a gap, an unsequenced gap. All this new stuff that we have now has no gaps. But also, I scanned through the entire genomes, count up every single letter. You can have other letters like R or Y. That means that, like, for every potential combination, it's like we know it's an A or a T, there's a letter they put there. We know it's a C or a G, there's a letter they can put there. We know it's not a T, well, there's another letter they can put there. So all those other variables, a lot more than 4 possible letters. Once you have only A, C, G, and T, that's only 4 possible results. So you can take that and turn it into binary, where an A can be 0, 0, a C can be 0, 1, a G can be 1, 0, and a T can be 1, 1. And so I did that. Now, I've used binary before. In fact, even with GAPI data, you can just call the N 0, 0. And if there's 10,000 N's in a row, you know there's a gap there. But for this brand new data, I mean, it came out 6 months ago, I guess. Brand new chimpanzee, fully sequenced. They used a long-read sequencing technology, and it was so good that they're actually able to know which of the 2 chromosomes in each chromosome pair they were sequencing. And since these are captive bred animals, they know the paternity and the maternity of the animal. They were able to say, okay, this is the father's chromosomes, and these are the mother's chromosomes. So essentially, they kind of sequenced 2 different chimpanzee genomes at the same time. Very interesting. So I'm working with this data, and I'm going to say, well, I said, I'm going to convert this to binary. So I did. And now I've got a file that's literally 1/4 the size. And in memory, it takes up 1/4 the amount of memory. Because most letters for most systems are in, I don't know, sometimes they're different, but at least older systems, 1 letter on your screen takes up 1 byte of memory. So there's 256 possible letters. Now, we have a lot more than 256 possible characters, so it's usually more than 1 byte per letter on the screen or in memory. But for real simple ASCII data, which is A, C, G, T, periods, question marks, all this normal stuff on your keyboard, there's 256 possible letters with the escape character and the return character and stuff like that. And A, C, G, and T have a specific 8-bit representation in memory. But with binary, if there's 2 bits per letter, I can shove 4 letters into 1 byte. So it's 1/4 the size. That's great. So I started doing that. And I had to write some code to be able to say, okay, I want to go to this position in the genome, in the binary genome. So I said, okay, I have to convert it from here to here to here, and here's where the chromosome starts. Had the list of all the chromosome start and stop points in the binary genomes. And then I had to be able to convert from binary to regular nucleotides and regular nucleotides into binary. And I got all that working, and it works beautifully as far as I can tell. One of the problems I have as a programmer is I'm very often off by 1. So if I say, okay, give me from this position to that position, I might start 1 letter shy or 1 letter too far, or end 1 letter too shy or 1 letter too far. And I had to do a lot of ground truthing. But I did a lot of work on this, generating all this data. And then I realized that I never ran a control. Whoops. And when I ran the control, I realized that all my data was worthless because I had plus or minus 1 errors all over the place. What I did was, I said, I need to be able to do this, but compare something to itself. So I took chimpanzee genome, the 1 copy, the father's copy of the chimpanzee genome, I think, and I said, okay, here's BLAST. I would pick a random chromosome and 1,000 letters in a random place on that chromosome, and then I would use this computer program to find a match in the genome that I took the sequence from. And it would tell me 100% match, 1,000 letters long, 100% identity, perfect. And it would tell me a position. And I would go and try to pull the position, and I realized I was usually off a little bit, one way or the other. So back to the drawing board. I got all that prototyped out, got it all fixed, and then I started running more data. But they also had a gorilla genome and a bonobo genome and 2 different orangutan genomes, one from Borneo, one from Sumatra. and a gibbon genome. Did I get them all right? I think that's all of them. So I got all these different great ape genomes with super high quality, every one of them, and I can do testing of those to themselves, those to others. And so I compared the human genome, the chimpanzee, human to bonobo to gorilla, both orangutans and the gibbon, and then chimpanzee to the other copy, gorilla to the other copy, bonobo to the other copy, and then the others also. And I'm generating all this data, and I'm starting to realize a couple of very interesting things. One, given that the human and chimpanzee genome, even if they're 98% identical, they're not, but let's just say they are, that means you get, on average, 1 letter difference every 50 letters. 2 letter differences per 100 is 1 out of 50. Now, nothing in biology is random like that. You expect clusters of differences and maybe some long stretches in other places, but most all the matches were about 50 letters-ish. And yet, most of the time, they came from the wrong chromosome. I mean, if I've got a section of chromosome 1 in human, it's supposed to be comparative, whatever that word is, to chromosome 1 in chimpanzee, then when I take a piece of the human chromosome 1, it should co-locate on chimpanzee chromosome 1, but it usually didn't. In fact, okay, I'm going to say half the time. I did a heat map just to see how often they hit on the right chromosome, but maybe half the time on chromosome 1, the other half at a random location in some other place in the chimpanzee genome. And that's telling me that even though, let's say it this way, it's telling me that I can have this 1,000 letters in the human genome, but it doesn't match the corresponding area. The chimpanzee genome, there's a better match somewhere else. So the true match isn't even being found, and a false match is being found. So most of those 50-letter results actually are spurious. Most of them are less than that. I also noticed very quickly that about 17% of the time, it would come back with no result. As in, here's 1,000 letters of the human genome, and it could not even find the minimal length, which was 22 or 24 letters. I think it was 24. I'm going to guess 24. Something like that. Anyway, it could not find, 24. The shortest possible match was 24 letters out of 1,000, 17% of the time. That's pretty bad. Now that means that there's a lot of differences between the human and chimpanzee genome. And yet every once in a while did find a nice long match. Sometimes 200 letters, 300 letters. I did find 1 or 2 in the 2,000, 2,500 range. So that seems to be probabilistically about the limit. Now I haven't scanned the entire genome. I don't know that there's not a longer piece out there. The longest piece that I did find after looking at like 5 million letters, well, yeah, even with the mistake I made earlier, where I was off by 1 or 2, it's still about 2,000-ish letters, even with all the other ones added in there. So 5 million-ish letters, and let's say the longest match is 2,500. I'll have more on this later. One of the reasons why I'm reporting this now is because it's a long-term project I had to keep on putting on the back shelf, because I got a lot of other things I'm having to do for work and for biblical genetics, and this programming was taking over my life several times. I just had to stop, Rob, put it aside, go brush your teeth, or something like that. I had to, basic life still has to happen. And so there's something else happen though. When I do chimpanzee to chimpanzee, I get about 10% of the time it kicks back with no match found. That's odd. Same with gorilla, same with gibbon. So this algorithm, this program, is failing to find a match when you do one individual in the species to another individual in the species, but a lot less often than when you do from one species to another. So I can't say that 17% of the genome between humans and chimpanzees is not a match. But I can say that there's some extra percentage of non-matching between the two genomes. Now it's going to take a lot more work to actually figure that out, to ground truth it all, to look at every single time I get a hit and find out where it is. And the other problem is, in these genomes, they're not loaded in the UCSC genome browser. So I can't type in a location and find what gene that I'm looking at when I find a big match. I mean, they're there somewhere, but I can't use that because the genome browser is using an older version of the genomes. What I could do is take one of those matches, then go online and use the online blast, just search the chimpanzee genome for a match, and that will tell me the location. Then I go UCSC genome browser, look at all the genomes, all the features and stuff like that. I just haven't quite gotten there yet. So I really got myself in too deep on this one, but it was fun at the same time. But it was, I mean, grueling hours, late nights, early mornings, weekends. Every moment at the airport where I could sit down with my computer and connect my spare drive, I was programming, and I was trying to crack these numbers. Now, what I need to do is, instead of randomly picking places, I just need to scan down a chromosome. Maybe chromosome 2 of human, because that's where we supposedly had the fusion between those two ape chromosomes to produce our chromosome 2. That'll be very interesting. But I already know that a lot of those matches are going to go to the wrong chromosome. I probably also, though, need to relax the stringency a little bit. And so instead of looking for perfect matches, allow for gaps and allow for less than 100% identity, because that'll help it stick to the proper location. And once I do that, I might be able to pull some of them and then count up differences and see which the longest match is. But this is just an exploration as part of a project where I'm looking to finally figure out what is the percent similarity between humans and chimpanzees. Now, I got to work on this because in July I'm giving a presentation on this at the CRS conference this summer, the Creation Research Society conference. So I got a lot of work to do still, and this is just part 1. In the end, though, this doesn't have much application to the creation evolution debate. How long the longest stretch is, honestly, even in the evolutionary scenario, that depends upon many, many, many things. What's the mutation rate? What's the recombination rate? And what's been the population size historically? Those things are going to influence this number that I'm trying to generate. So it's just one number that we can put on the chart, and then we have another number, another number, another number. In fact, for this debate on how similar the genomes are, there are 3 numbers we need to pay attention to. One is, for the areas that we do have in common, what's the percent similarity? Well, that's going to be pretty high. It's going to be on the order of 96, 97%. And we already know that. But for the areas we don't have in common, when you roll those into the equation, the percent similarity is going to drop a lot, which is why Richard Buggs in the UK is saying the percent similarity is around 85, 86%. That's why Jeff Tompkins at ICR has been saying that the percent similarity is a lot less than the evolutionists want it to be. And I know the evolutionists are trying to slam his reputation, saying he doesn't know what he's doing, but he's holding his own as far as other people analyzing his stuff. And but we're all making mistakes here. Every single party involved in this has been making mistakes. So a little bit of humility might be good for us all. But that's the second number. So what's the percent similarity of what we share? Then what's the overall percent similarity is the second number. And those 2 things are critically important. We have to separate them. But then the third number, this is the number that's actually the most important. It's how many mutations must have occurred in our history to generate this number of differences. That's a totally different question. Can the mutation rate be used to explain what we see between the 2 genomes? I'm guessing no, but it's a type B experiment, because if it can be used to explain the difference between our 2 genomes, well, then we're left with well, maybe God created it that different. And that's why the evolutionists say 6 million years, because that's what it takes to accumulate that many mutations. Don't know, but the 3 questions: what's the percent similarity of what we share? What's the overall percent similarity, which will be much less? And then how many mutations? But the mutations depends upon a couple of things also, because you can have a single letter difference, single mutation changes an A to a G or something like that, fine. But you can also have macro mutations, giant deletions, giant inversions, giant insertions, duplications, and those sorts of things will change a lot of letters all at the same time. And yeah, so in other words, if you want to actually answer that third question, you're going to have to develop a very sophisticated mutation model. Yeah, and good luck with that. That's going to require a lot of assumption. Yeah, some ground truthing and populations and things like that, but it's going to be really, really, really hard to build a model that's actually realistic, because you're going to have little changes and big changes and everything in between happening over millions of years. You're going to have the loss of most mutations, because almost every mutation is lost just randomly. Something like 99.9999 something percent of all mutations are expected to be lost. So to drive the number of differences between the two species, what's the mutation rate? That's really what I want to know. I don't know if I'm smart enough to get there. I don't know if I have a long enough lifetime to get there. But maybe one of you, maybe you're working in a graduate project, maybe an undergraduate research project, and you want to start cracking into that question. That'll be fascinating, because maybe there's some low-hanging fruit for someone to find. I don't know, just possibly. Well, that's all I had to say. That was a long soliloquy. It's actually, I'm a bit out of breath trying to walk and talk at the same time. Never done that before. I hope the camera's not been too jiggly for you. I've got the thing focused on my face and it's been tracking my face, but I'm also holding the camera in my hand on a stick, so it's a little bouncy. But hey, I hope you like science as much as I do. I don't think you'd be watching this if you didn't. If you are a creationist and you're struggling with what the other side is trying to say about what you believe, just relax. There's a lot of unknowns, there's a lot of uncertainties, and there's a lot more work still to do. And very often they've made these giant pronouncements that turned out to be not true. And so some of the modern pronouncements that they're making right now, in fact, some specific ones I'm supposed to be working on that I'm going to get to, I'm kind of hoping that those will be easy to chip away, or maybe even just sweep away entirely. In fact, I had a guy post a rather inflammatory comment on one of my videos I just posted on Facebook. And I'm like, dude, just wait to July, because my new paper in the Journal of Creation is going to come out, and I'm answering that question exactly and explicitly. And when you read that paper, you realize you won't be able to say what you just said. In fact, he lifted something from one of Richard Dawkins' books, and it's absolutely answerable scripturally, and I can't wait. I'm not going to tell you what it is yet, because I can't yet, because it hasn't been published. But it's just one more answer to help us say the Bible is factually true, the Bible is historically true, and evolution just doesn't quite work the way the evolutionist wants it to work. So thanks for listening. Really appreciate it. Thank you so much for my small group of people who's helping sponsor my program. If you would like to help contribute, just look down the links below. My list of supporters is growing, and I really, really, really appreciate it. You are totally encouraging me to keep on going. Because you know what? I need encouragement. I'm trying to encourage all of you, but I need encouragement too. It is hard to get insulted, to be belittled, to be laughed at, to be called names. You know that. I know that. We're in this together, working hopefully for the glory of God, hopefully for the furtherance of his kingdom, but hopefully to grab some people who are vacillating and encourage them. The Bible actually is true. And if it's true, then you take a very solid look at the claims of Jesus Christ. Just saying. You all have a wonderful day. I'm going to turn around and work back. I've walked, this is 1.5 miles, so I'm going to have a lovely stroll back without talking, just thinking about what I said. Hopefully I don't have to retract anything. But I'm going to have I don't know, half an hour more to think. God bless you.